Good morning, madam. I am Jinanda Prasad, second year biotechnology. This is the group video seminar. We are two members, me and my friend Naresh. We are taking the lead in this video about antibodies and what is antibodies and types of antibodies. We are continuing. My friend was continue the session.
Good morning, madam. My name is Naresh. I am studying in second biotechnology at Spaces TT College. Today, I want to explain about antibodies. First of all, what is an antibodies? The antibody or immunoglobulin or Y-shaped glycoprotein present within the serum and other body fluids. And the common function of antibodies when the virus or antigen or other microorganisms when enter into the body and they recognized by the antibodies and they killed by the antibodies and the structure of antibodies and the I am explain about structure of antibody. The structure of antibody composed of two different chains. One is called light chain and another one is called heavy chain. And the light chain about molecular weight of 25,000 Daltons. The light chain consists of two types of polypeptides and one is called lambda polypeptide and another one is called kappa polypeptide. The lambda polypeptide is in human beings present within the 60% and the kappa polypeptide in human beings antibodies present within the 40% and the lambda polypeptide are present within the mites only 95% is up to 95% is present within the mites in lambda. The kappa polypeptides only 5% are present within the mites and the other one is called heavy chain. The heavy chain or light chain, these two chains are composed with each other but in each other with disulfide bridges and the structure of antibody present within the two different regions. First one is called FAV region and second one is called FC regions. These two regions are combined with each other is called Hing region. Their upper part is light chain and heavy chains. Upper part is composed of amino group and lower part is composed of carboxylic group and they are present within the antibodies. And these light chain and heavy chains are binding with adjoining of disulfide bridges with non-covalent interactions. And let's see about structure of antibody. See, we explain about structure of antibody. Structure of antibody. The structure of antibody consists of Y-shaped molecules. This is called heavy chains. These two chains are heavy chains and it is called light chain and another one is called, this is also called light chain. And these light chain and heavy chains contribute each other binds of disulfide bridges. These disulfide bridges are non-covalent interactions between two light chain and heavy chains and The upper part is antibodies are composed of amino group both of light chain and heavy chain, and the lower part is carboxylic group both light chain and heavy chain. And in these surroundings or this is called FAB region. FAB region is called, FAB means fragment of antigen binding, and another one is called FC region. FC region. And the FC region is called fragment of crystallization, and the antibody, it is light chain. This light chain about 25,000. This light chain about 25,000 daltons in molecule. These light chain are composed of two different polypeptides, and they are, one is called lambda and other one is called kappa. And the lambda polypeptides are in human beings composed of 60%, and another one is called kappa. The kappa is called in human beings composed of 40%. In mites, the lambda is present within the mites above up to 95%, and mites, the kappa is present within the mites only 5%. And these two heavy chains are composed with each other and binds with each other of only disulfide bridges. And the FAB region or ring region, connect with each other only. FAB region or FC region connect with each other only ring region, and it is also amino group. And it is light chain and the heavy chain. Another one is called heavy chain. The heavy chain composed of molecular weight is 50,000 daltons, and it is structurally composed of 5 types of polypeptides, and they are, one is called alpha polypeptide, and other one is called gamma, and third polypeptide delta, and mu, and epsilon. These 5 polypeptides belongs to heavy chain. These 5 polypeptides are composed of among these 5 polypeptides are in alpha, delta, and about 330 amino acids, and it is derived into CH1, CH2, and CH3. And the mu and epsilon heavy chains contain a total 440 amino acids, and it is divided into CH1, CH2, CH3, and so on. And it is structure of antibody.
First of all, types of antibodies, that is different types. There 5 categories that are existing, arranged in a manner: IgG, IgM, IgA, IgE, IgT. I'm explaining about one of 3: IgG, IgM, IgA. First of all, IgG, immunoglobulin gamma antibodies are mostly abundant immunoglobulins present in the serum. The serum concentrate are accounted about 80 percentage of the um, uh, 80 percentage of the 80 percentage of the total in the serum immunoglobulin occurs in the concentration. The IgG antibodies are subsequently divided into four types because of an amino acid sequence. Amino acid sequence are arranged in their heavy chain modulation, and the four types of IgG: IgG1 65 percentages, IgG2 23 percentage, IgG3 8 percentage, and IgG4 4 percentage. In IgG, antibodies are mostly abundant immunoglobulins. That's all. In this IgG 1, 2, and 4 are the mostly useful in the antibodies are protecting and developing the other more features in the IgG antibodies. And next, IgM. IgM means um, immunoglobulins mules. Immunoglobulins mules. IgM antibodies. Immunoglobulins mules. So, okay. IgM are constituent about 5 to 10 percentage of 5 to 10 percentage of total immunoglobulins present within the serum or occurs in the concentration of, first of all, IgM contains two different types of categories, that are monomeric IgM and pentameric IgM. The monomeric IgM contains, the molecular weight is 1,80,000 Daltons present within the serum of B lymphocytes and acts as a receptor. And the other one, pentameric IgM. In the pentameric IgM contains 5 monomeric IgM, and each of the monomeric IgM and that enzymes or anything are covalent each other to binding the and recognition in the serum. And the most of it in this the addition of the any molecule or any enzyme conducted with J means joining chain, and the arranging of IgM has pentameric and monomeric substances are the pentameric and monomeric antibodies are arranged in such a way of two types, FC individual and FAB individual. And IgA antibodies. IgA means immunoglobulin alpha antibodies are constituent within the serum. About 10 to 15% of total serum immunoglobulins occurs in the concentration. The IgA immunoglobulin are there are two types, IgA present within the serum, and IgA are present within the extracellular secretions such as such as breast milk, saliva, tears, mucus, etc. And the two IgA antibodies secretions usually exist in the dimer. What is a dimer? It means dimer means two mono. The dimer were classified into two monomers IgA antibodies isolated within the covalent or non-covalent bonds are occurring to the IgA IgA antibodies, and some other joining cells, J chain cells, also present to combine the strand in uh, antibodies, and the antibodies are called as secretory antibodies in IgA antibodies. Okay, thank you, giving me this wonderful opportunity. We conclude the session.